It's a real pleasure to have Meredith Rocca speak. Usually at LRIG we focus on technologies, but fitting with the theme today, we brought in a card-carrying toxicologist to address some of these issues of why we're seeing that theme and trend of more antibody INDs versus small molecules. And Meredith will touch on that. It's a real pleasure to have her. She just very recently was nominated and confirmed to be on Jerry Brown's Reproductive Tox Committee. So we're really happy to have her. Meredith?
Good afternoon. Can everyone hear me? I know I stand between you and the wine and between me and the wine. So I will be happy to discuss this in more detail at that time. So I'm going to keep this pretty high level since this is not necessarily what this group is most focused on. So I'm going to tell you a little bit about background about biologics and why they're different, what you need to do for toxicology studies for them, a few considerations on the design, and just sum up. Okay, so general principles of toxicology apply to biologics. This should not be a surprise. So what is the purpose of doing tox studies? Many people feel it is to find out what are the toxic effects in rats. But personally, I don't care what the toxic effects are in rats when you come down to it because we're developing drugs for humans. So what we're trying to do here is to characterize the toxicity, looking for potential target organs that are translatable to humans. We're looking at dose exposure dependence, and we're also looking to see if any effects that we do have are reversible after the drug is removed. And so we use these to predict what's going to happen in human pharmacokinetics, what might be a safe first-in-human dose, and what parameters we can monitor in the clinic. If we have a drug that is causing problems with the spleen, that's nothing we really have any good biomarkers or clinical way to monitor. On the other hand, if our biologic is affecting liver enzymes, for example, that's something that is routinely screened for and will make it much easier to develop that drug. The other thing you need to remember about biologics, and that's where this first-in-human dose comes in, is that because of their long half-life, traditionally, they are developed in patient populations. That you're not starting out with your healthy volunteer single dose, because that single dose that you're giving is going to remain in their system for months and months and months. And so typically, although it wouldn't necessarily have to be the case now that there's more data on biologics, that's how it's been done. And so if you're going into a patient population, chances are you're also not going into a single dose, because what you're developing this is something where you would like to see a response over time, and if you have patients, you would like to give them a fighting chance to have this benefit them. So typically, you're going into patients, and you're going into repeat dose studies in patients. So what's different about biologics? Well, they don't call them large molecules for nothing. Just to get an idea of the differences within biologics, obviously something like aspirin is very small. I don't know if you can read on my print up there, it's about 180 Daltons. But then as you begin to get into some of the proteins and then peptides, you're getting up into, for insulin, it's about 5,000 kilodaltons. For Neupogen, which is a protein, it's 18. But then when you get into monoclonal antibodies, you're talking about something that's probably going to be about 150 kilodaltons. And that is going to affect lots of things about how this drug is developed and handled by the body. Okay, so the first thing is they're large. The second thing is the metabolism. One of the things that people are constantly working on is tweaking the small molecules so that the metabolism will not give you that reactive metabolite, or so that your metabolite will be similar in your different populations of people, that you won't find one population metabolizes it differently than another. That's not an issue in antibodies. They are just catabolized to proteins and broken down to peptides, and there are no metabolites to worry about. So that also makes your tox easier in a way because you don't have to worry about which species has the same metabolic profile. There is none. Specificity. The big draw for antibodies is that they are very epitope specific. This is typically considered to be a very good thing so that you will get less off-target interaction, but can have some other issues as well. We'll get to that. Immunogenicity. This is different than small molecules. Obviously, you're putting a foreign protein into a living system, and you may well see immunogenicity. And just because you're putting a fully human protein into a human does not mean that you're not going to see immunogenicity. I have seen almost no molecules that don't give you at least a little bit of immunogenicity in some small percentage of the population. Okay? And to make it a little more difficult, if you do see immunogenicity in your animal models, you have to know how to deal with that, but it doesn't mean you're going to see that in your humans, and vice versa. I've seen ones where there is absolutely no immunogenicity in the animals, and you do see some in the humans. So it really isn't predictive, but you've got to deal with it. The big point of biologics is people believe that the toxicity is often due to exaggerated pharmacology. Well, that's what we're looking for. We're looking for something that is very, very targeted. And so with our toxicity studies, when we're giving an abundance of these drugs, it's not surprising that we may get exaggerated toxicity and pharmacology from that. So, for example, I worked on a drug that is supposed to increase red blood cell count. Well, you increase red blood cell count enough, it's toxic. So it doesn't mean that it's not toxic, but it was on target. So for toxicology studies with biologics, there is a completely separate guidance from the International Conference on Harmonization, the S6 safety guideline is specific to just preclinical work for biologics. And there's recently been a revision to that. So it kind of comes in two parts. So just like regular tox studies, you should be trying to perform your studies in 2 species, but they must be pharmacologically relevant. And if you don't have 2 species that are relevant, 1 species can be sufficient. And this is very different than what you'd see for a small molecule, in which you may have numerous different species, depending on the type of test you're doing. In terms of the duration of your study, just like most small molecules, you need to always have your studies be longer than what you intend to put them into humans. But remember, I just told you, most first-in-humans are already long multiple-dose studies. So you may well be doing longer studies right off, but you're probably going to do many fewer of them. The other thing about biologics is that there is a limit, and this is part of the recent revision of the S6, is that the maximum length of tox studies for biologics is about 6 months. So that's significantly shorter than you might have to do for a small molecule. So, the relevant species issue. To be relevant, a species must be pharmacologically active, is what it tells you. There was a time when they thought that using animals that didn't necessarily have the target would show you off-target toxicity. And it's pretty well accepted now that using targeted biologics in a species that doesn't have a target is a waste of time and money, and if anything, will just give you results that you can't properly interpret. Therefore, what you need to do early on, and this is very important when you're trying to develop the antibody, is that you need to know for your target species or your tox species, where's the target expression? Is it going to be similar in humans as it is in the animals? What's your homology? Remember, most of these mAbs are very, very specific, and so if you don't have good homology, you may not have good binding, or the binding affinity may be very different. The other thing is the function. that all functions are not conserved. So even if we do have a receptor, say in the rodent, that this binds to, its signal transduction pathway and function may actually be very different than it is in humans. And you need to demonstrate function. And that gets a little difficult in many cases because what we're trying to do is have something that's so targeted and we're blocking our receptors most of the time, that really what you're trying to show is that you don't have a function. And it's hard to prove a negative in some cases. Okay, so this is your basic laundry list of things you would do for any toxicology program, with a couple additions. So the first thing is species relevance. You always have to have a relevant species. You can't just pick any non-clinical animal off the shelf and decide that's what you're going to do your tox species in, even if you have a small molecule, because you also need to have systems that are very similar and you need to be concerned about metabolites, et cetera. But for biologics, there is a higher hurdle to show function. So something that's typically been done is something called the tissue cross-reactivity test. And this is an immunohistochemistry test, which is done using all the human tissues that exist in duplicate and all of the animal tissues for the species you're considering using in triplicate. And so you stain every tissue you can imagine. There's a list of 37 of them. And see, is your binding similar in your target species as it is in your humans? These are done on cryosections. And the reason there's a question mark there is that these days there are certainly more sensitive methods than immunohistochemistry to tell where our target expression is. We also have the literature where many of these targets have been very, very extensively studied, and we know where they are in humans. And so if we can show that they're in the same areas by PCR or some other method, that may be perfectly acceptable. And that's one of those things that's sort of changing at the moment. However, when you go to your first IND and they ask you where your tissue cross-reactivity package is, don't be surprised, because that's something that they're used to seeing. You're going to do single and repeat dose toxicity studies, most likely, like you do with anything else. You need to look at your kinetics, anti-drug antibodies, and also local tolerance, because most of these are going to be given parenteral. These are not oral compounds. You do have a responsibility to consider genotoxicity and carcinogenicity, developmental and reproductive tox testing, local tolerance, as I said, and other studies. You may need to do some mechanistic work if you really are on target and you're seeing this exaggerated pharmacology. You may need to do some additional studies to explore that and explain why it's toxic. Safety pharmacology is a requirement for all molecules, but with biologics, typically these studies can be included as endpoints in a repeat dose study. So, for example, a monoclonal antibody is not going to get into an IKR channel. So you would not be so concerned about perhaps cardiovascular toxicity from that exact channel, and you certainly would not do a hERG test because it would just gum up your entire piece of equipment. So that's your basic. So how do you then go on to design your study? Well, just as in clinical studies, the dose is always the issue, right? So generally, you want to use the clinical route of administration. This is not unusual. However, feasibility is allowed to be considered for biologics. Because, my example here, if you have a drug that's for IV injection, it may not be feasible to inject a mouse weekly for 6 months with your drug. That they have teeny little veins in their tails, which is how you would do that with a mouse. And it's just well known within the regulatory groups as well that this is just not a feasible thing to do. So therefore, you may want to use a different route. You may be able to put it in an indwelling catheter and do it as an infusion. You might be able to do it intraperitoneal, a sub-Q. But you'll need to, at any rate, understand your PK and bioavailability. But at least they are reasonable about the idea that some of these things are not feasible in animal models. Dosing frequency. Ideally, exactly what you do in the clinic. However, we know these have outrageously long half-lives. And if you're only dosing once every 3 months in the clinic, you could do your 6-month study with 3 doses. And that probably is not going to be enough to get the level of drug in the system that you want and the level of comfort the clinicians are going to want, because you're going to be dosing these patients perhaps for years, and you would like to get numerous doses into them. So in that case, you may dose much more frequently. The other thing that will happen is your PK may be very different in your tox species than you expect in humans. In humans, your nicely well-behaved monoclonal antibody will have a half-life of about 3 weeks, give or take. However, in a mouse, because they metabolize these down much quicker, it may only be a week. So in that case, you may want to dose weekly to keep up with what would be normally a once every 3 weeks dose in a human. Dosage. We would like to be able to show a dose-response relationship. You would like to be able to show some toxicity and also a no toxicity level. This can be very challenging, that many of the targets that we are after, that if we're going to block a certain receptor, which we're concerned about because it's on a tumor, you can give those tox species all of that drug that they can possibly put in them and perhaps never see any toxicity at all. That's where it's really important to show the function and to show that you have done your due diligence that this is a relevant species. Once again here, feasibility is a consideration. The dose volume can be limiting. So, for example, you start at your clinical supply that you think it's going to be 20 mg per mil given IV. Well, what is the maximum amount of monoclonal antibody you can actually put into a cynomolgus monkey at one time? And depending on the lab and the type of thing you're doing, it could be between 5 and 10 milliliters. But that gives you a high dose of 200 milligrams per kilogram. So typically, that's a pretty whopping high dose. And this is where the difference really comes in between small molecules and monoclonal antibodies. If I tell my team, my development team, that I need, so many grams of drug to run my 2-week tox study, generally they can make that on the bench. However, if I'm now running a full-blown multiple-dose monkey studies where I have to give 200 mg per kg every week, it's going to take a long time to develop that much antibody. And you ideally want to use something that's as close to the clinical formulation as you can, because you don't want to do these studies again. You want to do them once and do them right. So this is where there's frequently a time delay. However, in the most recent revision of the regulations, this limit dose idea is evolving. So that according to the M3 guidance, which applies to both large and small molecules, you can go with about a 50-fold margin over what you expect to be your highest clinical dose. So even if you're seeing no toxicity, if I can say I have dosed those animals with 50 times more than a human is ever going to see, then that's usually acceptable. Now, is that acceptable for biologics? It has not been completely tested yet. One of the things they do talk about, however, in the biologics guidance, is that you can dose to maximum pharmacological effect. And so in many cases, your pharmacokinetics are going to plateau. You're going to get to steady state after a while. And not only that, but if you're trying to go after, say, a particular ligand or receptor, if you can show that you have already saturated all the binding for those, then those are acceptable reasons to limit your dose. And if none of those work, they do say that for biologics, you can do 10x over the highest clinical dose. But as I said, none of these limit doses have really been tested that I'm aware of at this point. That we toxicologists really like to see toxicity because that helps us do all those things we talk about. Choose a safe dose, figure out what we can monitor, understand how we can translate our animal into our clinical model. And when we don't have any, particularly regulatory authorities get a little nervous about that. So we'll see whether or not they will follow their own guidances. Yes, the regulatory world is a little different than where most of you folks operate. So, dealing with immunogenicity. So, administration of monoclonal antibodies may result in anti-drug antibodies. And you can have several different effects on your kinetics that typically it's going to clear your drug out of the system. That's normally what happens. You're going to look at your kinetics, and you're going to see that your drug levels are just falling right off the cliff. However, that doesn't have to happen. Sometimes your antibody can actually sustain the antibody in the circulation, and so that you actually may have longer PK. And then the thing that's most important is, are they neutralizing? If you have an antibody that's to a section, so this is your ADA, this is your immune system reacting to the monoclonal antibody, if it binds to a part of the antibody that's not involved in the function of the antibody, it may be perfectly fine if you have lots of antibodies there, because your drug may still be working and doing exactly what you need it to do. So there are strategies to deal with this, and this is where a lot of issues come into tox studies. You can change your route of administration. If you're giving something subcutaneously or IM, you're much more likely to get an immune response, think vaccines, than you are if you're just giving something IV. And in a case like that, that may be a reason why you say, this is the appropriate route, because I get higher exposures and it's more representative of what we're going to see in humans. You can change the frequency of administration. That you'll hear people talk about dosing through. In a case like this, if you keep a high enough antibody level constantly there, the immune system does not react the way it does to a vaccine, where you purposely let the response mature, where you give a vaccine and then you wait, and then you give it again, and you wait, and you give it again, and you wait. If you don't do that, if you keep it very high, sometimes you can avoid that sort of immune response to begin with, and then when all else fails, you just increase the dosage. And usually you can give enough drug that even if it does produce some antibodies, you still have enough active drug on board that you can show that you're doing things. Now, this is just kind of an aside for toxicity warning, is that if you get an anti-drug antibody that cross-reacts with an endogenous protein that's essential to be there, you have a very, very serious problem, because you probably can't turn off that immune response. So there is an example of that. And that's with an EPO-type drug that's to increase red blood cells. I don't think it's the one that Lance was using. But one of the formulation changes that was made in it that seemed very minor, what it did was that it did induce ADAs, and those cross-reacted with the people's endogenous EPO. So anytime you go after a target where you're giving something that may cause ADA to a endogenous protein, you have to be very, very careful. These people who had these reactions ended up with pure red cell aplasia, and they will need to have transfusions for the rest of their lives in order to live, because they cannot make their own red blood cells. Okay, this is the part that Michael likes the best. When we compare what we would be doing for, say, the single and repeat dose tox studies. So if you're a small molecule study, you're going to start out probably with a single dose. Then you're probably going to do a 1-week study so you can understand how high a dose you can go, that they're going to want to know what's the maximum tolerated dose. And in most of these cases, you expect that some of your animals are going to die. Then you move on, once you know more, to your 1-month study. You look at all the histology from that, try to understand what your target organs are, try to understand whether or not you can take the same doses into your next study, or will it be too toxic if you continue? And then eventually you get to your chronic 9-month study. And frequently there are many other studies in between those because you're trying to understand the specific toxicity. You've changed the formulation, you've done something to tweak it. Whereas with biologics, typically, the ones that I'm working with, there's going to be probably a single-dose study that's non-GLP that's done early on in development because you want to understand the PK and tolerability of your drug. But you're probably going to do this, as I said, with the PK group, and just it's going to be just kind of a general look-see. But from there, you generally move right into a repeated dose study because that's what you're going to need to move into the clinic. And depending on how long your first dose study is going to be in the clinic, you need to tailor this. So in the example I'm giving, we did a 1-month study which involved giving me 5 doses. And then from there, went to a 6-month study. I had one drug program in which the one and only standard toxicology study was a 9-month monkey study. No single dose, no 1 month, no 6 month. This was something that it was a second generation. We felt we understood the target well enough and our drug well enough that we didn't waste any time. We did one study, and it was acceptable. The other difference, of course, as I said, is you're going to want to use 2 species for both if you can. Typically, with small molecules, you're looking at rats and dogs. For biologics, frequently, but not always, you're looking at using non-human primates as the one that will probably have the epitope that's going to match what you need for the function. Now, that is something that can be screened for. And so, while you're screening for all those other things, one of the things that can be screened for is whether or not it binds to the receptor of specific tox species. I have done tox programs in rats and rabbits, and it can be done for a biologic. It doesn't always have to be primates. Recovery period. Typically, you want to see if whatever toxicity that you have identified is going to resolve once you stop taking the drug. Pretty standard in all small molecules that you do this. For biologics, it becomes more of an issue because of your half-life. If you have a half-life of 3 weeks and you want to wait 7 half-lives till the drug is now out of the system, and that's when they're first out of the system, and then you have to do a 2-month recovery on that, I've had cases where the recovery period was longer than the dosing period because you're trying to clear all the drug out. Those are not necessary anymore. If you have nothing to recover from, or if you can figure out what the pathway should be, particularly with the exaggerated pharmacology issues, you can indeed do without those very, very long recovery periods now. And so that's something that's relatively new. Okay, we're through most of the bad stuff. Genotoxicity and carcinogenicity. People typically think you don't need to do those for biologics. That's not quite true. What you do is you have to give rationale for why you're not doing them. And they're very rarely done. So the in vitro ones are just not at all appropriate for monoclonal antibodies, and the ones that you would do in vivo typically are not necessary unless you're doing something where you're giving a growth factor or something that's going to be an immunomodulator where they're concerned. Now, for doing that carcinogenicity study where you have an immunomodulator, you would typically do a 2-year rat bioassay. And what you'll most likely find is that the rat's not a relevant model. So, in the end, one way or the other, they're almost never done. Okay? But you do need to check the box and consider them. For reproductive and developmental toxicity, there are the 3 basic studies that are done for most drugs: fertility, teratology or development, and looking at postnatal development. Fertility is generally looked at by doing histology on the reproductive organs in repeated dose studies. Generally, there is no separate fertility study ever run for biologics. For teratology studies, and this is all very recent that this has become accepted, monoclonal antibodies do not cross the placenta by simple diffusion. And in fact, they require a special FC receptor-mediated mechanism to cross, which is not active for the most part until the third trimester. So it's not as if someone who doesn't yet know that they're pregnant is going to be at risk from having one of these antibodies early on, which is always the concern. Typically, these sorts of studies are done before any women of childbearing potential would be in a study for a small molecule. But in this case, depending on your patient population and your indication, if they are required, it's not going to be required until the marketing application for the most part. So this is a real difference in that you're putting off this huge, expensive study by years into development. That most of our drugs we know are never going to make it. And so if we can defer the studies to when they make scientific sense, this is a good thing. And the postnatal studies, these are typically done in rodents where you're looking at learning and memory tasks. If you can't use a rodent model, then these typically are not done either. There is something new called the Monkey Enhanced Peri-Postnatal Study Design, in which if you do have a drug that is targeted at women of childbearing potential, and you know that the target is likely to affect development, okay, then in that case, you most likely will have to do a study. But now it is a study, and this has now become accepted by the regulatory authorities. So instead of it being numerous studies that take years and 3 times the number of animals, you can get all the endpoints into one study. Other studies, I said, local tolerance, you're generally looking at injection sites, but you're doing that all the time in your general tox studies, and so you'll just make sure that histologically you look at those. Now, if you're changing your formulations, you might need to do another local tolerance, but that's generally a single-dose study and can be done very quickly. I talked about the mechanistic ones where you're talking, if you're trying to look for what's going on with your species. Also very interesting for biomarkers. If you're looking for something that you can translate into the clinic, this might be a time to do a mechanistic study. And safety pharmacology, as I said, is generally included in the general tox studies. So, summarize here. Tox testing for biologics is not the same as it is for small molecules. It's very based on the mechanism of action and the pharmacology of what it is that your target is. You're concerned about your relevant species, and that's going to drive your study design and your endpoints. And no talk on biologics would be complete without somebody saying that you need to use a case-by-case approach. And with that, I will take questions, and as I said, I will be drinking wine if people want to talk to me afterward.